cytokinetic ingression. Vijay says he's happy to take questions throughout the talk, like other speakers. So if you have a question, please raise your hand. One of us will come and bring you the mic.
Thank you, Suri. Let me also start by thanking the organizers for the invitation, and it's been a very nice meeting, and it's always great to come back to ICTP. So this is a problem that is ongoing, and it's a collaboration with Masatoshi Nishikawa from Hosei University. It's primarily the PhD work of my student Aditya Rajput. I'm broadly interested in problems of morphogenesis. I mean, shape transformations occurring at the level of cells and tissues really fascinate me a lot. And I think a large part of biology has really focused on static shapes. And we think about chemistry, perhaps mechanics, but shape hasn't entered so much into the game yet. And so that will really open up a new set of problems is what I think about. So I see this as a problem of coupling between geometry, signaling, and mechanics, signaling particularly at the tissue level. And I would like to think about minimalistic physical models, which will perhaps teach us some of the physics underlying such self-organization phenomena. A large part of morphogenesis in cells and tissues is driven by this structure called the actomyosin cortex. So this is a thin film of polymeric filaments just beneath the plasma membrane of cells. And what you see is a gooey meshwork of filaments, actin filaments, molecular motors straddled there, some cross-linkers, and lots of other proteins which could be straddling through the surface of the cell as well. It's well known and well recognized from several years now that the dynamics that happens in the cellular actomyosin cytoskeleton is largely responsible for things like the division of a cell, the movement in a cell, deformations in a cell, and so on. And even at a much larger scale of tissues, it's well recognized that the actomyosin cortex plays a pretty large role. Of course, it's much more strongly regulated by signaling at a more macroscopic level. But I mean, what's more fundamental than cell division as an example of morphogenesis, right? I mean, something that we all see in the microscope. A single cell divides into two cells, and it's really a problem not just of segregation of genetic material, it's also a problem of dynamical geometry. And that's the problem that I want to address today. And to tell you that there are some funny things that happen here. Okay. So one of the first things that we've recognized from several years now is that the actomyosin cytoskeleton is crucial in this process. So here you see a labeling of actin, Lifac cherry, and you see a nice ring of actomyosin that comes up in the middle of the cell, and the contractile forces that are generated in the actomyosin cortex eventually cleave the cell into two daughter cells. So the picture is that you have the actomyosin cortex. Somehow, perhaps due to flows, perhaps due to other mechanism, a contractile ring is assembled. The contractile ring has these contractile forces by definition, and so therefore, eventually you get constriction. So that's classical cell division for you, right? So what's the physics of the cortex? I mean, how do I understand how to think about a hydrodynamic or a physicsy description of the actomyosin cortex? So like I mentioned, the cortex is a very dynamic meshwork. What it does is something which is atypical of other condensed matter systems. It continuously turns over in the sense that there is continuous exchange of material with the surface and the bulk. Filaments that are stuck at the surface, they depolymerize, monomers sitting in the cytosol go and polymerize. So there's a continuous churning over of material. So on time scales much longer than the stress relaxation time scales, which are typically like the turnover time scales, it flows like a fluid, like a viscous fluid. So that's useful. And if you look at the structure, like I mentioned, it's a polymeric meshwork of filaments. There are straddled motors sitting there. And each motor consumes ATP, as we all know, and generates these contractile forces. And that's essentially the mechanism for why the system is driven out of equilibrium. How do I write a physical description for such systems? Well, of course, one can start at the molecular level, write down detailed molecular dynamical models, and so on. But as a physicist, one would like to look at more coarse-grained descriptions, a hydrodynamic, a thermodynamic description. And the most apt framework for this is active matter. We've heard several talks about this. It's all right, I won't need all of these equations, but I just want to flash out that idea that the general description is based on movement, transport, of the density of the meshwork, of buried concentration fields of motors and so on. Basically mechanics, Newton's laws, generalized to continuum descriptions, force and torque balance, constitutive relations that tell you how stresses are generated by strain gradients, and also a new thing called the active stress. You could also have more complex degrees of freedom, for example, the orientational degrees of filaments. Perhaps the more important thing, like I mentioned, is the coarse-grained description of the contractile forces is essentially manifest as an active stress, and that's what really drives the system out of equilibrium. Okay, so is this a good and useful theory? It is. So you can take a simplified version of that, and let's say you take the C. elegans zygote in the very first cell division stage, and this is a classic experiment by Stefan Grill, where you can actually show that if you write down a coarse-grained 1D description for the flows that are happening from the posterior to the anterior side, you can do a particle imaging velocimetry, so get the flows out, write down a hydrodynamic theory by doing force balance and a simple viscous description for the constitutive relation, and you get a prediction that the gradients in the active stresses, contractility gradients, are the source for the flows. And that you can actually test experimentally. So you can measure flows, you can measure myosin, and you ask, let's say I assume some relationship between myosin and active stress, can I actually fit this theory? And lo and behold, you can. So you actually get a flow in real units, some microns per minute. So therefore, that sort of tells you that this is a pretty good theory at this level of description. Now, if the flow description is good enough, what can it lead to? Here's a nice way to think about how patterns can come up in the cortex, spontaneous patterns. So I have some regulator of concentration of the active stresses, say myosin. So C is the concentration field of myosin. And like I said, this is the very good description of the flows in the active myosin cortex. So there are active stress gradients drive flows. So the flows will affect the concentration field of myosin itself. In addition to simple diffusion and perhaps some simple linear turnover, you will have advection. So the advection velocity V is generated from the flow equation. The flow equation itself has gradients of C, and so therefore this is a coupled closed system. So you can ask what are the possible states of the system, and you can show that if the Peclet number is large enough, basically if your active flows can beat diffusion, then there is a nice linear stability calculation that you can do and show that indeed the homogeneous state is unstable. So numerical simulation in the nonlinear regime shows you very nice looking patterns of myosin. So the heat map is the myosin concentration and the arrows are the velocity field. You can generalize this to include 2 species or maybe some other nonlinearities and can get even time-dependent patterns to come up in the problem. Okay, so I want to emphasize these are genuinely mechanochemical patterns in the sense that if you don't have this fluid velocity active stress coupling, there are no patterns. It's a boring dead equilibrium state. Back to cell division. Okay, so I know to think about forces and stresses in the actomyosin cortex. I know to think about patterns. Cell division is also a pattern. So can I explain that? Okay, so classic examples of cell division where some sort of an asymmetric concept is classical asymmetric cell division that we all learn about. And I mean, again, the C. elegans zygote is a fantastic example of that. The very first cell division is an example of a classical asymmetric cell division. The 2 daughter cells are not the same. There are certain chemicals which get preferentially partitioned into one daughter cell compared to the other. And so what this does is to accomplish certain developmental processes like an axis establishment as well as a germline and somatic line segregation. This pattern of the cell division is driven by certain surface patterns of polarity proteins, power proteins that they're called. And turns out that the dynamics of these polarity proteins is also regulated by the flows that happen in the cytoplasm. Okay, so this is a problem that we worked on quite extensively, so I won't tell you the details, that's another talk. But it turns out that you can take this description that we had for the mechanics part of the flows, couple it to evolution equations for the polarity proteins and the mechanical force generating proteins. And you can show that once you can do a very quantitative experiments, get out the concentration fields, flow fields, and so on and so forth, and you can fit things, lots of genetic manipulations, you can extract the parameters very, very systematically, and you can actually predict the spatio-temporal evolution of the concentration fields as well as the flow fields on the surface of the cell. Okay, so therefore this sort of lends credence to the idea that patterning via mechanics can even describe such complicated processes, and we also glean some ideas that people knew intuitively from their experience, but the theory shows that very clearly, that it turns out that the homogeneous state is stable, the polarity state is also stable, so what does the triggering from one state to the other is some developmental signal that comes in there. It's a very nice, robust way and controlled way of making transitions in patterns. All right, so this is cell polarity, but we want to go back to cell division. And in particular, I want to highlight one particular kind of division that happens. So this is the same cell division of the first C. elegans zygote. So if I play the movie, you see the 2 daughter cells, okay, cytokinetic ring coming up. You can't see much in the DIC image. I label myosin and I look at the midplane. And what you see is the myosin ingresses more from one side compared to the other. Okay? So if you look at the division plane, that is, you look at the plane in which the division is happening, you see the cytokinetic ring the cytokinetic ring does not go symmetrically towards the center. Okay? So that's the asymmetry that is there. So is this a one-off? No, it's very repeatable. It always happens. Is there some signal doing that? It doesn't seem to be so. So the conclusion, the picture that we have seen so far is a bit too fast for me. The picture that we have seen so far is that if you follow the time course of the evolution of the cytokinetic furrow, you see something like this in the division plane. Okay? So the red is the earliest time point and the blue is the last time point. Why does it divide asymmetrically? Why does it ingress asymmetrically? And is this something which is specific to C. elegans? Turns out no. Turns out many creatures, particularly the ancient ones, seem to have asymmetric ingression. Sand dollar is a classical example. So there's a couple of movies are lifted off internet, Twitter. And this is a fantastic movie that really shows, well, this side doesn't even ingress. And you see a front of an ingression front coming from the right to the left, and something, again, similar in the C-star zygotes. There are caveats. There are other signals here. For example, you can see a wave of some chemical going around. But nevertheless, the idea that there are asymmetric ingressions doesn't seem to be specific to C. elegans. So perhaps there's some general physics to be learned here. Do we know the orientation, the ingression point, the asymmetry of the ingression point? Is that something controlled by a signal? Is it always deterministic or is it random? As far as we could make out from previous studies and our own experimental collaboration, there doesn't seem to be any signal. You repeat the experiment many times and every time it comes from a random direction. Does it depend on the fact that you've taken the embryo and put it between the glass plates and so therefore you squished it a bit? Well, you can take the embryo and put it upside down and image it in the vertical direction, and even then you don't see any signaling happening. So therefore, it seems to be reasonably random. We don't know for sure, but it looks like the spindle is not having any serious effect. As far as we can make out, the spindle is more or less in the center throughout the ingression phase. So possibly the spindle may or may not influence the asymmetric ingression. We don't know. But all these things we've repeated many, many times in the lab, and these seem to be robust and consistent. So this is work with Masatoshi Nishikawa. He's been quantitatively measuring these things very, very well, as something we've been doing for the past few years now. So you can look at myosin, do SPIM, confocal microscopy, several Z-stacks, assemble it, and you can very clearly see the asymmetric ingression of the ring happening in this movie. So if you look at the division plane, you see this very nice ingression in myosin coming up there. So you can take that, digitally cut it open, and so therefore now you can quantify things. You can ask for how much asymmetry there is in the myosin levels compared to on the leading side, compared to the lagging side, and so on and so forth. So you can get very, very quantitative. How do we think about cytokinesis, and in particular asymmetric cytokinesis? So there have been a couple of works. One of the very early works that I know of is this very nice work by Hervé Toulier from the French Paris group, where they actually look at a very nice circular domain, assuming azimuthal symmetry there. And if you impose some ring-like contractility profile and you track the deformation of the surface itself, you can actually recapitulate something that looks like a cell division pattern coming up there. But remember, this is with an imposed ring-like contractility profile. To explain asymmetric division, I know a couple of studies, not many. So one study is a continuum model, models by stresses, strains, and so on and so forth. But what the study does is to impose a asymmetric boundary condition and just models the division plane, imposes asymmetric boundary condition, and tracks the front. And there you indeed see an asymmetric ingression of the cytokinetic furrow. Another model tries to do a more microscopic picture with lots of microscopic elements, filaments, motors, binding, and so on and so forth. And in some level puts in non-equilibrium energy input into the system, no hydrodynamic flows, no large-scale flows in the system. So we'll come back to this model. But the point being that all of these require an asymmetry or a ring-like pattern to be put in by hand. Can we get it spontaneously? Is there a simple hydrodynamic picture that we can construct? Okay, so what we want to understand is if we can couple contractile patterns, patterns that gave rise to nice hexagonal patterns of myosin that you saw, but with shape now. And so if you can get a coupling between pattern and shape, perhaps the shape patterns will follow the concentration field patterns and maybe cytokinesis will come out. Okay, so that's the picture that I want to build for the rest of the talk. Okay, so one first approach is to take models like what we've been discussing for the concentration field and put it on some curved surface. Simplest is a sphere. So you put in something where you have a surface concentration field and a surface velocity field. You write down force balance on the surface of the manifold, and you can then put in a bulk fluid as well, mimicking the cytoplasm. And for the simplest case, you can think of the cytoplasm as a Stokesian fluid with some viscosity there. And you put in appropriate boundary condition. The surface flow gives you the boundary condition for the bulk, and the bulk stresses evaluated at the surface provide attraction force. So you can take this system and again do a linear stability analysis and show that indeed that there is a certain length scale, which is basically the ratio of the shear viscosity of the surface to the shear viscosity of the bulk, and that's a length scale. And it's really the competition between this length scale and the size of the sphere that matters. So in the limit where this hydrodynamic length scale is much larger than the radius of the sphere, you get an instability of the L equal to 1 mode in spherical harmonics. And if the other limit, where the hydrodynamic length is much, much smaller compared to the radius of the sphere, you get a L equal to 2 instability. So in pictures, this really translates to the following. If you have an instability like that, this gives you a polarity state. There is a spot of myosin that comes up there. And if you have a L equal to 2 instability, you get a ring of myosin there. Okay, so if you get a ring, then perhaps if you make it deformable, you should be able to get cytokinesis. You can put in a bending energy following Carham-Helffrich and do a linear stability analysis. And you can show what are the possible deformation mode that comes up at the the onset of the instability. And surprisingly, you find that the Peclet number, the critical Peclet number for the instability, doesn't seem to depend on the bending rigidity. Okay? That's a bit of a surprise. So you really need to go further into the nonlinear regime. And so therefore, if you want to go far away into the nonlinear regime, you definitely need a robust numerical method. Now, a classical way of solving such equations on any surface, the physicist's favorite way, is to write down generalized coordinates, some curved coordinates, and you do that, and then you try to see if you can solve them. You find you can't do it for anything other than a sphere, but then you try to do it numerically, and then you realize, oh my God, there are coordinate singularities. So there are lots of 1 by sine theta factors, and you can't solve them. Okay. So what we've been doing from the past few years, along with a bunch of groups all over the world, is to develop a method which is a completely extrinsic description, that is, you sit in the ambient space, but make sure that only the intrinsic quantities matter. So this is the idea of tangential calculus, which is well known in the mathematical community. So you have a surface, and I think about a unit normal vector at any point on the surface, and that gives me a very nice way of constructing what I call a tangential projector. This is basically something that tells me how the fields change in the tangent plane to the surface. What this does is to give me a computational way to construct covariant derivatives of fields sitting on the surface. Things like concentration fields and their gradients, or velocity fields and their gradients, and you can show this is the same usual physics of covariant derivatives that comes up. You also have the shape operator, which basically is something that tells you the curvature tensor there. And you go, I mean, it's well known, again, in the classical intrinsic description, you know the metric and the curvature tensor completely capture the surface. In this case, it's the normal vector and the curvature tensor that completely capture the surface. So if I give you a velocity field, you can work out the simple math and show that the normal velocity, normal component, normal unit vector, and the curvature tensor evolve that way. It doesn't matter. This is basically just some calculus, nothing more than that. So the physical idea is the following. So if I know that I give you a velocity, you know how to find out how the shape changes. I have a way to get velocity from the active patterns. How about using that velocity to deform the surface? Okay. So that's therefore our complete model. So we have a mechanochemical patterning mechanism, concentration fields, move, diffuse, turnover, force balance, constitutive relations, some viscous stresses and active stresses. And now I say that I'm going to couple this to a evolution of the geometry, the shape. So I basically say that I look at the normal component of the flow, and that allows me to move the surface. So I'm thinking of like a fluid-like surface. And I can also compute the surface descriptors in terms of the normal component, normal, and the curvature tensor. So once the geometry changes, obviously that will feed back into the pattern. So there's a coupled dynamics between pattern of concentration fields and geometry. I'd like to understand what are the kind of resultant patterns. So one of the first things that we got, of course, if you have very low activity, you don't get anything. It goes back to a sphere. If you start from a sphere, it goes back to a sphere, boring term. One of the first things that we got was really very nice. So we start from a sphere. with a uniform concentration of myosin, and you have this coupling between geometry and dynamics, and you just let it go. No other external input, no other signaling provided by hand. And there are regions in parameter space where you get a very nice cytokinetic ring formation and what appears to be a nice cytokinetic furrow leading to a cleavage. If you increase the activity, you make it more contractile, turns out you get cytokinetic furrow. Very surprisingly, you get an asymmetric ingression. Okay, so it's hard to see from the 3D plots, but you can look at the division plane, and in the division plane, you follow the trajectory there, and you see that on the left one, you have something which is more symmetric. On the right one, you have something which is definitely asymmetric. So that reminds you quite well with the experimental flow profile there. Okay, so the picture here is that there is a competition between asymmetries that come up because of active advection, forces leading to flows, leading to concentration accumulation, and relaxation, either because of bending or because of turnover. And so if you create asymmetries faster than you can relax, surely you can enhance asymmetries. Okay, so that's a phase diagram in the Peclet number, which is basically a measure of...
Quick question here, right down here. So in a couple of slides ago, you sort of showed how the...
Yeah?
Next one.
Okay.
You showed how, how the flows are coupled to the surface.
Yes.
So this is a very naive question. So sometimes you can have binding, unbinding like things.
Yes.
So here this is tight coupling effectively.
So this is effectively binding, unbinding.
Okay, but all the flows, okay, fine, perfect, okay, thanks.
Simple linear turnover. Okay. Yeah. So there are regions in parameter space, particularly at high contractility, where you can get very asymmetric ingression coming up there. So there's a phase diagram, and the phase diagram has the following structure. That if you look at the Peclet number, which is basically a measure of activity, and a length scale associated with the correlation of the velocity field, you can show that apart from the homogeneous state, you first get a ring-like state consistent with the earlier picture that we had, and then a further instability, you get a spot-like state consistent with the earlier picture, and a further increase in the activity gives rise to a ring-like state. And the higher the activity you have, the higher the asymmetry. Of course, you never get a perfectly symmetric thing, that's never possible. So you get something which is more and more asymmetric as you increase the activity. Okay. So this is all simulation, I mean, this is all calculation. Does it conform to something we see in real life? Okay, so here it goes. So a couple of things that we've been looking at are the following. So let's try to quantify the asymmetries that you would see in cytokinesis. So many of these are symmetric divisions, several of them are asymmetric divisions, but one thing that's been observed in a couple of studies is that the cytokinetic ring ingresses at a constant rate. So this is basically some measure of the length of the furrow, and you plot that as a function of time in bacterial systems, in sea urchins, across 4 species, also in C. elegans. And it turns out that there is a regime where there is a linear reduction of the size of the cytokinetic ring as a function of time. Okay, so we have that ready. I mean, we can measure the ring and see what comes out of the theory. And so you define basically something which is the base radius of the sphere, the initial radius that you started out with, and the instantaneous radius of the ingressing ring, and of course a symmetry parameter, which I'll come to in a second. And from the theory, we find that indeed you get a very nice straight line ingression of the radius of the cytokinetic ring. Okay, and ingression rate, which is basically the slope of those curves, increases with activity. Higher the activity, higher the asymmetry. All right, so this is measuring something which looks qualitatively there. Can we get something more? So it turns out, remember this story that I had told you long back ago, that the continuum model with an imposed asymmetric boundary condition actually recapitulated asymmetric ingression there, only in the division plane. So it turns out in this study, they also looked at quantitatively from experiments the measure of the cytokinetic radius as a function of time, as well as this asymmetry parameter as a function of time. So theory can capture something, but can't capture some other features. We can do the same thing in our simulations. And this I already showed you, the variation of the radius of the cytokinetic ring as a function of time. You can also measure the asymmetry parameter, and you do see something rather remarkably similar to the experiments. Now, it's hard to compare these two data because I don't know what is t equal to 0. I don't know where do I define my time, initial time, and I don't know how those calibrations. So how about the following? So there is some quantity R by R naught as a function of time. There's some other quantity, asymmetry as a function of time. I do a parametric plot. I ask, hey, if the radius of ingression is so much, what is the asymmetry parameter? I can do the same thing in my theory. And you can ask what happens, and this is what you get. So there's no fitting here. I mean, we just adjusted one Peclet number to be as close to the experimental values as possible. So what you're asking is basically, if the cytokinetic ring has ingressed so much, how much is the asymmetry? That seems to be reasonably well captured with the theory. Remember, I told you about this cytokinetic ingression and this slope, which is basically the ingression rate, and there is a maximum asymmetry. So I also told you that this rate increases with the Peclet number. So one can do the calculation at various Peclet numbers, and for each Peclet number, say you calculate the maximum asymmetry and plot it as a function of this rate at which it is ingressing. We get some plot that looks perhaps some sort of a straight line variation there. So what our collaborator Masatoshi Nishikawa did was to take C. elegans zygotes, do lots of genetic perturbations. In particular, he did MLC4 RNAi, which is a nice way to knock down the strength of activity. So you feed the worms lots of bacteria, and you feed them such that their MLC, they touch on the myosin light chain kinase pathway, and so therefore their activity of myosin goes down. So basically what it means, you're reducing the activity strength. In other words, you're reducing the Peclet number. So you redo these experiments and you measure, again, similar quantities like the ingression rates and the asymmetry and ask what you get in the experiment. Turns out he got something like that. Okay, so this is the same delta quantity that we are measuring in the simulation, and he has a measure of the constriction rate. Of course, he can get it in real units. Perhaps there's some correlation between the two things that's going on there. Okay, I'm almost at the end of my talk. We've possibly explained ideas of cytokinesis and even asymmetries in cytokinesis. I mean, it's natural to extend that to other deformable geometries in cell biology. Classic thing that many of us know, or examples of hydromorphogenesis and so on. So some things are cooking in the lab. Okay, so this is a summary. So what we have is a simple mechanochemical model. There's very minimal physical input. There's a lot of calculus, but that's all. And so this seems to lead to spontaneous cell division patterns. And surprisingly, it can also capture asymmetric division. And there seems to be some sort of a quantitative comparison with data, not in terms of the molecular concentration fields or the shape parameters. That's again something which is cooking. But in terms of coarser parameters like the amount of asymmetry, the cytokinetic ring radius as a function of time, that seems to be reasonably well captured within this model. It's easy to include things like you regulate them by signal. I don't know if cytoplasmic flows are important, but that's hard to do simulation-wise. Perhaps ordering of the actin filaments is important because we know that the cytokinetic ring has very nice ordered actin filament structure. Perhaps there's some effect of the spindle. I don't know. These are things that we are exploring. All right, okay, so that's the summary. So these are the people who did the work, Aditya, my student, and Masatoshi, a friend from a long time, and these are the people who fund me. Thank you.
Questions? I'll go for the first. Oh, actually, students first. You know, it's close to the end. Oh, former student. But I'm walking up to Joe.
Yeah, beautiful talk. I mean, this asymmetric cleavage, I mean, it looks like a crack. Is there some analogy there? It looks like a crack forms there's some breakage of symmetry and the crack forms and the crack goes to the other side. I mean, is that any kind of valid analogy to what's going on here?
I don't know, but my first guess would be probably not, because the membrane is fluidy. This is really taking 2 minutes time roughly to do the cell division process. On those time scales, it's more of a fluidy structure. I don't think it's like a solid in which a crack front is propagating there.
Nice talk. Behind all that complicated mathematics, is there a simple explanation for the asymmetry?
So like I mentioned, so there is the active mechanical flows that come up there. The flows naturally have an asymmetry because they're trying to increase the concentration in one direction. There are mechanisms for relaxation like turnover and bending relaxation there. So this competition is what leads to asymmetry. So if the relaxation is much, much stronger compared to the asymmetries of the contractile flow, then you get something which is symmetric, more symmetric. But if you relax the relaxation phenomena is much weaker, asymmetries are the thing that get amplified. And that's essentially what more or less our picture seems to bear out.
I'll ask a quick question. Beautiful talk, amazing work. My first thing is, so you sort of provided this very nice intuition for how patterns are formed on non-curved surfaces first, and you couple things together. If you take the basic non-curved model and you look at high values of PE,
Yes.
Beckley number, do you, for the kinds of algebra you have, do you start getting things that look like traveling wave fronts? Or
Yeah.
Okay, so is this sort of, and this sort of goes back to what Joe asked, is this sort of asymmetric thing a manifestation of something that looks like a traveling wave front? No, it's not that at all.
No.
Okay.
I mean, for the wavy thing, there's a different physicsy mechanism that comes up there. This is not due to wavy things. We can follow the full time-dependent patterns there. Maybe I can just show you that. Okay, so again, so this is something that we are trying to compare with experiments, and you can see that there aren't any of those wavy patterns. So from the experiments, we can compute the cross-section and reconstruct the surface from the experiment so that we have the full ingressing ring there. We can get quantitative measures of myosin flow, curvature, blah, blah, blah. We can see the similar things in the simulation as well. As far as we can make out, there are no wavy structures. Okay. We can follow the space-time evolution there.
A very naive question, but do you see this asymmetry in like non-spherical cells also in nature?
So for the simulation, we started out with something spherical.
Yeah, but
we can start with an ellipsoid. It doesn't matter.
Okay.
I mean, of course, the region of parameter space changes a bit.
Yeah, but if you take like bacterial cells that are not spherical?
Bacterial cells have a very rigid hard wall.
Okay.
That requires a different physics.
Okay, thanks for the beautiful talk. I have two fast questions. The first one, are you putting some asymmetry, small asymmetry in the beginning of the simulations?
No.
How does it?
Homogeneous initial condition plus some small random perturbations. Made sure that
okay,
there are
is
no
some
asymmetry.
small random perturbation.
Just like linear stability analysis.
Sure. And the other question is, I mean, as a function of the activity, you see that the asymmetry increases.
Correct. Asymmetric, correct.
There is not really a transition, right? There is also a small asymmetry when there is a
asymmetric
small...
thing you'll never get. So it's really a quantification how much asymmetric it is. And that seems to
doesn't become more unstable. I mean, there is nothing...
Not that we can find. I mean, see, the asymmetry seems to be a continuous function of the Beckley number.
Okay, thanks.
Here again. As for the ring formation, can I have an analogy with a bubble, a water bubble?
Mm-hmm.
When you blow it too big, you can see that there can be that kind of ring formation where it tries to be two bubbles, and there is a competition between the surface tension and the pressure inside. Can you make an analogy to that?
Kind of, but these are non-equilibrium systems, right? These are systems which have a throughput of energy all the time. Those are typically equilibrium systems. I mean, you could, for example, heat up the system and get things like Marangoni flows and so on and so forth. But these are systems which convert chemical energy into mechanical stresses. That's the active part of the thing. And that's what we showed. If you reduce the activity, the asymmetry goes down. At some level, it doesn't divide at all.
Question here. Very nice talk. What would you imagine that happens if you have some constraint, physical constraint to this division? For example, have some force, wall in one of the sizes.
I didn't
So it's
understand.
an
I hold the
external
surface
force.
I hold the surface fixed, for example.
Yeah, for example.
I haven't done that, but I guess the pattern would adjust to that. I mean, for example, we have done simulations where it's not a closed surface, but with a boundary condition that is put in there. The pattern will evolve and give you some shape consistent with that. That can easily be incorporated. Okay.
Any other questions? If not, let's thank Vijay for a beautiful talk and also all the other speakers.